Have you ever stopped to think about this hidden, ongoing arms race?
Okay.
It's happening all around us in nature, but we rarely think about it. A silent battle. Today, we're doing a deep dive into that fascinating and, yeah, sometimes deadly world of plant toxins, specifically how they impact large herbivores. We're really zeroing in on two key animal families, Cervidae, so deer, elk, moose, that group, and Camelidae, which is your camels, llamas, alpacas.
Right. And our mission today is to unpack why some plants are toxic, how these animals manage to cope, or sometimes, tragically, how they don't, and also how their unique biology and environments create these really different vulnerability profiles. The goal is really to give you the key insights to understand this pretty complex ecological challenge and why it matters so much, both for wild animals and for domestic ones, too.
Yeah, and this deep dive is drawing from a really detailed comparative toxicological analysis, plus other key sources. It gives us a good look at the physiology, the ecology, and importantly, the management of these phytotoxin risks. Okay, let's get into it. So, at its core, this relationship between plants and the herbivores eating them, it's ancient. It's this dynamic ecological arms race. Plants, they can't just get up and run away, obviously.
Obviously.
So what do? Well, they evolved something like 200,000 different chemical defenses. We call them plant secondary metabolites, PSMs. And these aren't just like waste products.
No, not at all. They're absolutely crucial for the plant's survival. They defend against herbivores, pathogens, and interestingly, they even help attract pollinators sometimes.
Huh.
And of course, herbivores haven't just, you know, stood idly by. They've developed their own sophisticated counter-adaptations. Things like behavioral avoidance, basically learning what not to eat, and also complex physiological detoxification mechanisms inside their bodies. This sort of co-evolutionary dynamic, this back and forth, is really central to understanding plant poisoning, or what we call phytotoxicosis.
It sounds like a chemical chess game playing out everywhere, a whole chemical landscape that these animals have to navigate every single day, like an invisible minefield. Okay, let's meet the players properly. On one side, Cervidae, true ruminants, white-tailed deer, elk, moose, reindeer. And on the other side, Camelidae, pseudo-ruminants, camels, llamas, alpacas, guanacos, vicuñas. And what's really striking is they live in vastly different places, right? Arctic tundra, arid deserts.
That's right, huge range. Yet they both face this universal challenge, plant toxins.
So how do they handle it differently? Let's start with the cervids, the true ruminants.
Okay, so cervids have that complex four-chambered stomach.
Hmm.
Rumen, reticulum, omasum, abomasum. The rumen is really the powerhouse here. It's this huge fermentation vat. Can be up to 8 liters in a white-tailed deer.
Wow.
And it's packed with diverse microorganisms. These microbes are absolutely essential. They break down tough plant fiber, and critically, they detoxify many of those PSMs before they even get absorbed into the animal system.
And their eating strategy plays a role too.
Definitely. Their foraging strategy often involves eating small amounts from a wide variety of plants in one go. They're basically spreading the risk, not concentrating on one potentially toxic source.
Okay, now let's contrast that with the camelids, the pseudo-ruminants. How's their system different?
Well, they have a 3-chambered stomach.
Yeah.
C1, C2, and C3. Now, C1 and C2 function similarly to the ruminant rumen in some ways, but C1 has these unique glandular saccules and different motility patterns. It leads to really thorough mixing.
Okay.
But here's a really crucial difference. Camelids have a significantly longer retention time for solid food particles.
Longer retention time. So the food stays in there longer.
Exactly. And that longer period allows for more extensive microbial fermentation and potentially more detoxification.
Yeah.
They're also incredibly selective feeders. They use that narrow muzzle, that prehensile split upper lip.
Right. They can be quite precise.
Very precise. They pick specific plant parts, even stuff that's dry, prickly, salty, or bitter that other animals might avoid.
So these big differences in digestion, how directly does that translate into handling toxins? Does one group has a clear advantage?
That's the key question, isn't it?
Mm-hmm.
And these fundamental differences in anatomy and physiology, they will directly influence how each family processes and responds to plant toxins.
You'd think it might be straightforward, but this whole issue of phytotoxicosis, plant poisoning, it's a significant threat, but often kind of underestimated, wouldn't you say?
Oh, definitely underestimated. It's not just about animals dying acutely.
Right. It's more insidious than that.
Exactly. Those sublethal effects, they're often way more pervasive. Think about livestock. Abortions, deformed offspring, poor weight gain, less milk, photosensitization, chronic illnesses that just shorten their productive lives.
That sounds like a huge impact.
It is. The estimates are something like 3 to 5% of cattle, sheep, and horses on western U.S. rangelands are affected by poisonous plants every year. That's significant economically and for animal welfare.
And for wild populations like deer, it must be even harder to track.
Much harder. Diagnosing it is tough. Animals often die in remote areas. They get scavenged quickly. So you might have a large die-off attributed to disease or malnutrition when actually toxins were the root cause. And the risk isn't static either. It gets amplified by human pressures. Things like habitat degradation, which limits the safe food options.
Makes sense. Fewer choices.
Right. And introducing non-native ornamental plants into suburbs. That presents novel threats. These animals haven't evolved defenses against garden plants from halfway across the world.
Like you, maybe.
You is a prime example. And then add climate change into the mix. It's changing where plants grow. It's changing their chemistry, sometimes making them more toxic. The whole risk landscape is shifting under their feet.
Okay, so let's dive into the actual toxins, the specific chemical weapons. You said the concentration varies a lot.
Hugely. It depends on the plant part leaves versus stems versus flowers, the growth stage, environmental conditions like drought.
Yeah.
It's incredibly variable, but we can group them into classes.
All right, let's hear about some key classes.
Okay, well, alkaloids are a big one. Found in things like yew, larkspur, locoweed, ragwort. These are potent, often targeting nervous and cardiovascular systems. For instance, pyrrolizidine alkaloids, PAs, in senecio plants cause chronic liver damage over time. Taxine alkaloids in yew, as you mentioned, cause rapid cardiac arrest, sudden death.
Scary.
Ditropiny alkaloids in larkspur cause neuromuscular blockade, basically paralysis leading to respiratory failure. And indolizidine alkaloids like swainsonine and locoweeds cause that neurological condition, locoism.
Okay, alkaloids. What else?
Then you have glycosides. These are sugar-bound toxins. The toxicity is released when that sugar part is broken off, often during digestion. Cardiac glycosides, like cardinalides in oleander or foxglove, directly mess with the heart's pump, causes arrhythmias, heart failure.
Oleander. Isn't that common in landscaping?
Extremely common and extremely dangerous if ingested. Then there are cyanogenic glycosides found in plants like chokecherry. They release hydrogen cyanide.
Cyanide. Like in spy movies.
Pretty much. It halts cellular respiration, stops cells from using oxygen, leads to rapid death, often with this characteristic cherry red blood because the oxygen isn't being delivered.
Wow. Okay, what's next?
Oxalates. Think helogeton, greasewood, even rhubarb leaves. Soluble oxalates bind up calcium in the body. This causes acute hypocalcemia, low calcium and kidney failure, because oxalate crystals precipitate out in the kidneys. Skunk cabbage is another one with oxalates, but it mainly causes intense mouth pain, which usually stops animals eating enough to get systemically poisoned. A built-in deterrent.
The lever or any others?
A couple more key ones. Resinoids, like cicotoxin, found in water hemlock. This is a potent central nervous system antagonist. Causes violent seizures, rapid deaths from respiratory failure. Very nasty stuff. And finally, granotoxins. These are found in rhododendrons, azaleas, laurels. They basically lock sodium channels open in cells, causing persistent stimulation.
What does that look like in the animal?
Clinically, you see profuse salivation, vomiting or regurgitation. That's a really key sign in camelids, by the way, colic, and various heart and neurological problems.
So many different ways for plants to be toxic.
It's incredible chemical diversity.
Yeah.
But what's really useful for vets is the toxidrome concept.
Toxidrome?
Yeah, it's basically a syndrome, a recognizable pattern of signs associated with a class of toxins. So even if you don't know the exact plant immediately, if you recognize the toxidrome, say, the signs of cardiac glycoside poisoning, you can predict effects and start treating the underlying problem.
That makes sense. Treat the symptoms based on the toxin type.
Exactly. It guides the initial response.
Okay, let's go back to the animals. Cervids deer, elk. You mentioned their foraging strategy is key, eating a little bit of lots of things.
That's their primary defense, yes. Spreading the risk, avoiding a toxic dose of any single plant. It's why people sometimes think deer are immune to certain poisonous plants, the so-called refractory myth.
Ah, so it's not true immunity.
Not physiological immunity in most cases, no. It's a very sophisticated behavioral adaptation. They do eat plants like false hellebore, skunk cabbage, water hemlock plants, highly toxic to others, but carefully. Small amounts, mixed diet.
But that behavior isn't foolproof.
Definitely not. Its Achilles heel is environmental pressure. Nutritional stress, harsh winters, droughts can force them to eat more of those toxic plants just to survive. They get less picky when starving.
And the human factor. Those ornamental plants.
That's the other major vulnerability. Introducing novel, often highly palatable, and highly toxic ornamentals. Yew is the classic example. It's an ecological trap because their evolved behaviors just aren't equipped to recognize this new danger, especially when other food is scarce. Leads to significant die-off sometimes.
Right. Yew seems like a big one.
It's one of the most significant threats, especially in winter. Animals are often found dead just hours after eating it right near the shrub. That rapid cardiotoxicity from taxine alkaloids
And finding yew leaves in the rumen.
is a key diagnostic clue, exactly. And while white-tailed deer seem a bit more tolerant, not immune, but tolerant, other species like moose, elk, mule deer are highly susceptible.
And diagnosing this in the wild is still really tough.
Inherently difficult. You rarely see them sick. You find them dead, often decomposed. So you're relying on post-mortem exams, looking at the scene, analyzing rumen contents, running toxicology tests. It's detective work.
Okay, let's shift gears to camelids, llamas, alpacas. Their situation sounds quite different because they're mostly domesticated or captive.
Profoundly different. Their risk profile is shaped almost entirely by that captive status, their natural selective feeding behavior, often totally constrained by fences and what they're given.
So the risk comes from us.
Overwhelmingly, yes. It's anthropogenic, human-caused. Poisonings often happen when pasture diversity is low, forcing them to eat plants they'd normally skip. But an even bigger danger is accidental exposure. People dumping gardening clippings, oleander, rhododendron into their pasture. That's a huge one.
Even dried clippings.
Yes, dried or wilted plants can actually become more palatable sometimes, and they're a common cause of fatal poisoning. Contaminated hay is another route. And young ones, crisis, they're particularly vulnerable.
So for camelids, risk management isn't about wild ecology, it's about good husbandry.
Exactly. It shifts entirely to responsible care and owner education.
Can you give some specific examples in camelids? You mentioned rhododendron.
Yeah, rhododendrons, including azaleas, often from discarded clippings. Signs in alpacas can include that profuse salivation, dehydration, their C1 motility slows right down, uncoordinated regurgitation, heart arrhythmias. Lab work often shows metabolic acidosis, kidney injury signs, electrolyte problems.
And oleander, you said that was really potent.
Extremely potent. Just a couple of leaves can kill an alpaca. It usually shows up as a triad. GI signs, anorexia, vomiting, slow C1 motility, cardiovascular signs, slow heart rate, arrhythmias, and acute kidney failure.
Sounds grim. Is treatment possible?
It can be, surprisingly. Prognosis is fair to good if you catch it early and treat very aggressively. Immediate vet care is key.
Interesting. Any others?
There was a fascinating case with Cotoneaster. A llama died from cyanide poisoning. Showed severe difficulty breathing, that bright red blood we talked about.
Yeah.
But what was really interesting was the clinical course seemed a bit slower than you'd expect in a true ruminant like a cow. The hypothesis: the cyanogenic glycosides were hydrolyzed, releasing the cyanide more slowly in the less acidic environment of the camelid C1.
Huh. So even subtle digestive differences change how the poisoning looks.
Precisely. It highlights how those physiological differences really matter in practice.
So wrapping up the camelid risk,
Yeah.
it's primarily human actions. Garden waste, bad hay, not clearing toxic plants from enclosures.
That's the bottom line for them.
Okay, so let's try to directly compare them now, this physiological face-off. Who actually handles toxins better overall? Is there a clear winner?
It's not that simple, unfortunately. Susceptibility isn't just one thing. It's the outcome of this whole integrated multi-stage detoxification pathway. And each stage can differ quite a bit between cervids and camelids, creating these unique vulnerability profiles.
Right, so we have the digestive differences. Cervids, rapid fermentation, high throughput. Camelids, longer retention in C1, potentially more microbial detoxification there.
Potentially, yes. That longer retention time might allow for more complete breakdown of complex PSMs by those foregut microbes.
And those microbes are really the first line of defense.
Absolutely. They're the primary detoxifiers for many compounds. They can degrade complex things, adapt over time, which explains why gradually exposed animals might tolerate something that poisons a naive one. Some microbes even break down tannins or turn cyanide into nutrients.
Incredible little chemical factories in their gut.
Pretty much.
Okay, but what happens if toxins do get absorbed past the microbes? Then it's the liver's job, right?
Exactly. Toxins absorbed from the gut go straight to the liver via the portal vein for biotransformation, breaking them down further. And here we see another critical difference.
Oh.
Studies indicate that camels, at least, seem to have lower activity of some key liver enzymes involved in detoxification, things like certain cytochrome P450 enzymes, or pseudocholinesterase compared to other domestic ruminants like cattle or sheep.
Lower enzyme activity, meaning their liver might be less efficient at handling certain toxins once they're in the bloodstream.
That appears to be the case. It suggests a potentially reduced capacity to handle certain xenobiotics, foreign chemicals, after they've been absorbed.
Yeah.
So it creates this potential Achilles heel.
So their foregut is maybe better at preventing absorption, but their liver is maybe less equipped to handle what does get through.
That's the potential trade-off, yes. It helps explain that paradox we mentioned. How can a camelid be so hardy, thriving on tough desert plants, yet be acutely susceptible to specific poisons in things like oleander? Their first defense line is strong, but the second line might have specific weaknesses.
Fascinating. Okay, so beyond their internal biology, external factors play a huge role too, the shifting landscape of risk.
Huge role. Plant poisoning risk is definitely dynamic.
So what are the big environmental drivers?
Well, habitat degradation and nutritional stress are major ones. If preferred food is scarce, maybe due to overgrazing, land conversion animals are forced onto lower quality, often more chemically defended plants.
Hunger makes them less picky.
Exactly. And malnutrition itself can trigger pica, that's a depraved appetite, where they might compulsively eat poisonous plants they'd normally ignore.
And climate change. You called it a threat multiplier.
It really is. Rising temperatures are changing where plants grow, potentially exposing animals to toxins they've never encountered. Drought and heat stress can also directly increase the concentration of toxins. Alkaloids, tannins, cyanogenic glycosides in certain forages can reach lethal levels.
So the plants themselves become more dangerous.
Sometimes, yes, and higher CO2 levels can increase carbon-based toxins while diluting protein content. So animals might have to eat more of this lower quality, potentially more toxic foliage just to meet their nutritional needs. It's called compensatory feeding, and it can push them over their detoxification limit.
Wow, a complex cascade of effects.
It is. And then there's the human-wildlife interface. Expanding development brings those non-native toxic ornamentals, yew, rhododendron, oleander, right into cervid habitat, plants they have no natural avoidance for.
The ecological traps again?
Exactly, especially attractive in winter. Plus, agricultural practices matter. Herbicide use can sometimes make toxic weeds more palatable as they wilt, and contaminated hay for confined animals completely bypasses any selective feeding behavior.
Okay, this paints a pretty challenging picture. So what can actually be done? How do we protect these herbivores?
It requires a multifaceted approach, definitely. For livestock and camelids, good integrated toxic plant management, ITPM, is key.
What does that involve?
Things like smart grazing management, proper stocking rates, avoiding known toxic pastures during high-risk periods, maybe providing nutritional supplements so they aren't forced to eat bad stuff. Also, control methods, mechanical removal, targeted herbicides, but a really critical caution with herbicides. You must remove animals from treated pastures until those plants are completely dead and dried up. Wilting often makes them more appealing temporarily.
Good point. What about when an animal is poisoned?
Then it's clinical management. Prompt vet intervention is crucial. First step, remove them from the source, obviously. Then supportive care, IV fluids for dehydration and electrolyte balance, activated charcoal to try and bind toxins still in the gut. And as we mentioned, for some specific toxins like cyanide, there are actual antidotes.
Okay. And for prevention, especially for people with llamas, alpacas, or other livestock?
Prevention for owners comes down to education and vigilance. Know the risky plants, ornamentals, common weeds. Regularly inspect pastures and fence lines. Secure fences well. And the biggest message, never ever feed garden clippings, lawn mowings, or tree prunings. That is such a common and often fatal mistake. Also, inspect your hay carefully for contamination.
Seems like straightforward advice, but critical. What about managing risk for wild deer?
For wildlife managers, it's more about landscape-level solutions. Habitat management is huge. Improve and maintain high-quality habitat to reduce that nutritional stress that drives them to eat risky plants. Public education is also vital. Inform homeowners near wildlife areas about the dangers of toxic ornamentals, encourage planting native, non-toxic species, and strongly discourage feeding wild deer.
Why discourage feeding? People think they're helping.
It often does more harm than good. It concentrates animals, which spreads infectious diseases like chronic wasting disease. It can cause serious digestive upsets because the feed isn't natural. And it degrades the surrounding natural habitat. It's generally counterproductive.
Right. Okay, so wrapping this up. The core insight seems to be that plant poisoning risk for both deer and camelids is this really complex interplay between their unique biology and all these dynamic environmental and human pressures.
That sums it up well. And if you compare them directly, for wild deer, their risk is largely tied to the health of their ecosystem and how well their evolved behaviors hold up. For captive llamas and alpacas, the risk is almost entirely defined by the owner's knowledge and husbandry practices.
It really makes you think, doesn't it? What hidden ecological traps might be sitting right in our own backyards or neighborhoods, affecting the wild animals we share space with? And maybe, what simple things could we do to reduce those risks?
That's a great question for everyone to consider. And scientifically, there's still so much to learn. We need more comparative research on those gut microbes using metagenomics to see the whole community, and more on the liver detoxification pathways, the SIPOMI. Plus, understanding exactly how climate change is altering plant toxin profiles is critical, and we really need to look more closely at the sublethal effects of chronic low-dose toxin exposure in wild populations. That's often overlooked.
So, the research continues.
Always more to learn.
Well, we've only scratched the surface today on this incredibly intricate, really vital area of ecological toxicology. Hopefully this deep dive encourages you, our listeners, to keep observing the world around you with maybe slightly different eyes, connecting those dots between the plants, the animals, and the environments we all share.